Awesome. Let's go ahead and get started. Welcome, everyone. Thank you for joining me here today. This is going to be mostly presentation, part workshop on closing gaps in alternative protein science. Many of you may have attended my talk yesterday that kind of gave an overview of why it's important for us to rethink our meat production systems at all. So I won't recover that kind of terrain. And today we'll kind of dive straight into the three alternative protein science pillars and focus specifically on cultivated meat research advancements and example white space opportunities. So let's dive right in. Just so everyone here is operating from the same level of knowledge, my organization is called the Good Food Institute, and we are a nonprofit think tank focused on accelerating alternative proteins. We do so through different aspects of the change-making process, both engaging the public, private sectors, and also ensuring that we're making substantial scientific and technological progress as a field. I sit here on the science and technology team, and this team is pretty much focused on identifying white space opportunities. We exist to generate ideas for where we are today, where we need to be in order for these various technology segments to succeed, for alternative proteins to succeed. And then we mobilize the funding and mobilize the people and institutions to solve for those white space opportunities. So we'll be talking a lot about some of those opportunities in the cultivated meat domain today. But first, I just want to make sure everyone has a shared understanding of alternative proteins. So when we talk about alternative proteins, we mean how can we produce meat that people love, that tastes exactly like conventional meat, eggs, and dairies, but utilizing plants, animal cells, and microbes. So that means that we split the field of alternative proteins into several technology segments. The first is plant-based, the second is cultivated, and the third is fermentation, kind of this very underexplored pillar. So first for plant-based. I won't dive into this a ton, but essentially plant-based meat, egg, and dairy utilize plants and fungi as inputs in order to produce meat that fully recapitulates the sensory experience of animal meat. So you basically have plants and fungi as inputs, and your end product might look something like this. And the core research areas involved in plant-based meat production are, first and foremost, crop development. What kinds of plants are we utilizing for plant-based meat development? And then ingredient optimization. Have those crops been then optimized for a plant-based meat application? A good example of this on the crop development side is soy, right? Most of our plant protein in the plant-based meat space comes from soy, some wheat, some pea protein as well. But soy has really been optimized as a feedstock for livestock to eat. And it hasn't really been optimized for human consumption and not been optimized for a plant-based meat application. So how can we explore different kinds of crops and then optimize the existing crops we have, like soy, wheat, and peas, for this kind of end application in mind? And how can we get the right ratios of fats and oils and sugars and binders in the plant itself, and optimize each of those for a plant-based meat application as well. And then this third category of product formulation and manufacturing, that's largely about texturization. Like, how can we devise like creative new production methods like Burgett Decker's shear cell technology, or 3D printing, or experiment with different formulations for assembling these end products so that they resemble whole cut muscle products and not just the kind of unsophisticated cuts of meat that we see today. So this thing pictured here is a plant-based steak. It's a fungi-based steak produced by Meaty Foods, and they estimate that this will become commercially available in 2021. So this is really produced just with a fungal scaffold. And some plant-based meats might also look like this, right? We can use plant proteins and basically convince them to behave like animal proteins. And then for cultivated, this is what we'll spend the bulk of our day talking about. So I won't dive into too much of it right now, but the essence is that we're utilizing animal cells and cell culture methods in order to grow genuine animal meat directly from the cellular level. And this is a product from Aleph Farms, an Israeli-based cultivated meat company. And then lastly, we have fermentation, which I think is a hugely exciting and really, really underexplored research area. And we're really excited to start kicking off research and innovation in this space. But essentially what fermentation is utilizing microbes to produce ingredients for alternative protein products and sometimes biomass for alternative protein products. So an example of an ingredient is here what's pictured is kind of a protein powder that's produced from utilizing basically microbes to suck carbon dioxide out of the air and then utilizing them to produce complete proteins, essential amino acids that can then be incorporated into alternative protein products. This is a company called Air Protein that's really building a circular economy by utilizing those waste streams. And then this is a picture of Thomas Jonas from Nature's Find, formerly known as Sustainable Bioproducts. And he found, he was on a team of scientists commissioned by NASA to go to Yellowstone National Park and basically find an extremophile in the volcanic springs. And they ended up finding a microorganism that was 50% protein and a complete protein with all 12 essential amino acids that also happened to have a very muscular filamentous structure that was very meat-like in texture. So they've basically used fermentation, a novel fermentation method, to grow a lot of biomass, essentially grow a lot of those microorganisms, and they assemble them into these meat-like sheets that you see here. So fermentation also has a lot of opportunities that have been underexplored that I wish we had the time to cover today, but perhaps in a future talk. But I think a thing to note about fermentation is that microbes are just the most efficient possible processors of anything, pretty much, compared to plants and animals. They're just far more efficient. They don't need to sleep. They don't need sunlight. They work round the clock. They eat pretty much anything. So there are real opportunities for us to produce sustainable processes through fermentation. So in the last couple of years, really last 5 years since this field has started kicking off, we've seen substantial progress in research advancements, in commercial successes, and yet substantial white space remains. We need so many more scientists and engineers focusing on closing gaps in alternative protein science. And like I mentioned yesterday during my talk, students really play a pivotal role in building the alternative protein field. These white spaces that we identify over the course of today and beyond, some of them are really well suited for students to tackle, both when we talk about research ideas and commercial ideas and ecosystem level interventions like getting courses set up and things like that. So today we're going to focus mostly on cultivated meat, on understanding the state of research in the various technology sectors or core research areas associated with cultivated meat science. And we will talk about a few examples of white spaces that exist in this space. But before we dive into that part of it, I do want us to have a really brief brainstorm as a group, and to kind of understand where you all are at, get your brains churning around what you think the central bottlenecks are to cultivated meat being successful, and what you think you can do as students in order to help overcome those bottlenecks. So I will share the link to our Miro board now, and it would be lovely to get everyone else plugged in on this. So if everyone can just open this link, if everyone can just open this link on their respective computers, we can join in on this Miro board and get our brainstorm started. And people should really feel free to hop off mute so that you don't only have to hear my voice all day. Can you give me a thumbs up once you have the Miro board open? Okay, sweet, so it's working. So let's take a brief moment. A central idea that I want to land across, even though the remainder of this talk won't be about this, is that students, exactly where you are today, at your university, studying whatever field of study you're studying, can do a lot in the way of advancing alternative proteins. So let's do a bit of a brainstorm and surface some of those ideas. The whole of this brainstorm will be 10 minutes, but the starting point is, and let me share my screen again. Loving seeing everyone's cursors here. What is your field of study? So maybe take a minute and go in and just write on a Post-it note what you're studying right now so I can get a better sense of the varying perspectives in this room. If anyone is having any problems with Miro, let me know and we can troubleshoot. Awesome. I'm temporarily hiding these cursors. Yeah, I see biotechnology popping up. There's engineering. In case anyone is having trouble typing in the Post-it note, you can just kind of double click on a Post-it note and start typing. I see computer science popping up a couple times, materials engineering, sustainable business, infectious diseases, FAF, which I don't know what that is. If someone wants to hop off mute and explain what FAF is, that would be great. I see geology, biology, mathematics, synthetic biology, video game development. Very cool. Oh, very big sticky note for biotechnology and economics. My background is also in economics and global health. And psychology. Okay, very, very cool. Thank you. So now that we have kind of an accounting of the different fields of study in this room, I would love for us to take just 2 minutes, either quietly or you can shout out answers as you come up with them. But from your perspective, knowing that this room is full of people who either specialize in alternative protein science, are brand new to alternative protein science, and might vary widely in terms of whether you know a lot or a little about where we're at, and you offer very different perspectives in terms of the fields of study that you specialize in. Let's take just 2 minutes for us to brainstorm together. From your perspective, what are the biggest barriers to cultivated meat success? And I'll start a timer here on your Miro board. Ooh, I'm loving some of this. Yeah, this is great. We'll keep this brainstorm going for another minute, but I see some themes emerging. All right. Any last ideas? I think I see the I see someone has um, drawn a really great frowny face, which I love. And yeah, I think I see kind of ideas popping up a little bit more slowly around the board. So why don't we stop there? Um, and yeah, talk about some of these ideas. So I think a big grouping that I see here is consumer acceptance. Like, will the public be able to switch? I see status quo bias. Yeah, cost or consumer acceptance, labeling battles, and um, public perception. Maybe food safety also kind of falls into this bucket, concerns around food safety. I see a couple things surfacing around ethics. Is it as ethical as vegan food? Really curious actually to hear about this. Do the people who put that answer down want to hop off mute and explain what you mean? I will take that as a no. Okay, so we have questions potentially around whether or not these alternative protein products are actually animal free, and I think that's an interesting topic of discussion. We can definitely surface some of those insights later on in this presentation. Jump to more than minced meat. Yeah, I see that as an idea here. Can we create more sophisticated cuts of meat? The regulatory aspects of things, will labeling battles and label censorship continue? Can we actually get regulatory acceptance of these kinds of products? Can we actually scale production? Can we move beyond the bench scale and the pilot scale and achieve a large enough scale and a low enough cost to actually sustainably feed the world, so that alternative proteins are not just a pipe dream. Um, and then there's some questions here around costs of cell culture medium potentially. That's absolutely right. Achieving maybe price parity, specifically to chicken, and things like that. Yeah, the structural complexity of animal flesh. These are all really, really great. And I do think we've touched on basically all of the biggest barriers to cultivated meat success here. Um, and I love this last sticky that I just saw, not enough people working on it. So if we don't get enough research funding, if we don't get enough scientists and engineers focusing on overcoming some of the technical bottlenecks that we just identified, that could be a real barrier to the viability of cultivated meat in the long term. Great. Thank you guys for your ideas. So with all of this in mind, knowing that all of this spans various disciplines, and you all as students are kind of uniquely positioned at the university to potentially tackle some of these questions, or to create the ecosystem necessary to tackle some of these questions. Why don't we move to the third phase here of our brainstorm? What can you do as a student to help overcome these barriers? I will put 2 minutes on the clock, and you can go ahead and start. Fifteen seconds left. Some of these are really fantastic. All right. Thank you. Yeah. So you guys are making me laugh. I love some of these ideas. I'm assuming it's Thomas there from KU Leuven who wrote GFI student group. That's absolutely right. You can absolutely start a GFI student group. And that's something that we talked about at length yesterday. Happy to speak more about this later. Yeah, you can help create more high quality, publicly available research and guides. I love that idea. Like, as students, is there a way maybe to create 101 level protocols for cultivated meat that make it easier for people to understand the technology? Can we make it so that students with little life science background can actually grow meat themselves? I see a lot here around research, absolutely. Research advancements are very needed in the university realm, and you can do a lot to persuade existing academic labs to take on research projects. You can absolutely take on research projects of your own. Tell purist vegans that they're wrong. I think that's a really great answer. Yeah, in alternative proteins, we work really hard to basically solve the question from the supply side, right? It's less about convincing people that they need to eat less meat and eat more plants. And it's more about like, hey, we can give you exactly the meat that you love, but just produced a much better way. It's cheaper, more affordable, it's tastier, and requires fewer inputs. It's much better for the environment. So that's kind of the theory of change. Study a relevant area. That's fantastic. I can give you a list of priority disciplines involved in alternative protein science. Love this idea around kind of socializing alternative proteins, normalizing consumption within our immediate social groups, and getting courses started, adding it as a university module or course for chem bio disciplines, absolutely. Trying to get the department to offer courses, outreach to professors, all of that is really fantastic. And we definitely have some resources that can help you accomplish any of these things. So awesome. I'm going to stop my screen share again, come back to the main room. Thank you guys for your ideas, and mostly for I think landing home the central theme, which is that like you are exactly as you stand, incredibly valuable to the state of alternative proteins. And there's a lot that you can do from your place in the university. Also, Nana, loving the unicorn. Okay, I'm going to pause here really quickly before I dive into a presentation around cultivated meat research advancements um, and some example white space ideas. Are there any questions that people have at this stage? I'm just seeing right now this contribution from Michael in the chat as well. Yeah, there's definitely this question of long-term versus short-term. Consumer acceptance might have a ton of variables. The like causal chain is really long and hard to understand. Absolutely, but there's a lot of research progress we can do now to advance the foundational science, so that the thing, where can you find a list of priority study programs? What a great question. Let me surface that link for you right now. So this is a list of key disciplines for alternative protein science. It's pretty exhaustive, but at some universities, it's called a different thing. Yeah, Aditya, it's often hard to know what to work on given the secrecy in the industry about what we already know how to do. That's right. Aditya is one of our student leaders at Wageningen. So if you have questions about running a student group, he'd probably be a great person to talk to. Yeah, I agree with you, Aditya. I think it's important for us to generate as much open access knowledge as possible. And maybe one of the biggest white spaces is how can we get companies, yeah, sorry, Aditya Vase, Frederick? Yep, thanks. How can we get companies, how can we align incentive structures so that companies share more information and they're working together to advance the field rather than just advancing information on a proprietary level? I think that's a great project for a student to think about. Like, is there a platform we can build to make alternative proteins follow more of a GitHub model, right? Like open source information. I think that would be really cool. Yep, there's a question here about computer science listed as a potentially relevant discipline. CS is absolutely relevant to alternative protein work, and that's because computational sciences are really, really needed here. So I realize that computer science is quite broad, but there's an example of how computer science and computer modeling is really important to cultivated meat in particular is the Cultivated Meat Modeling Consortium that you can see at the cmmc.org, and we'll talk about a little bit later as well, at least briefly. But essentially, like, instead of going through the really expensive iterative process of designing bioreactors and bioprocesses, can we take a miniaturized bioreactor and model it and model cell behavior within it at scale? Can we, instead of characterizing individual plant proteins, can we use bioinformatics to do that at scale across the entire plant kingdom to find the traits that we value in plant-based meat? There's some really great questions here in the chat that I do hope to get to at the end of my presentation, but I'm going to pivot now kind of back to this presentation that I know a lot of y'all came for. If I can find my presentation at all, let me, oh no, I've lost you guys. I literally don't know where the Zoom went. Give me a moment. Okay, there you are. So share screen. Okay, so I promise we'll have time at the end to get to some of those questions. Thank you for engaging in this very brief brainstorm. Now we're going to kind of dive into the various core research areas involved in cultivated meat and a discussion of those like example white space ideas. And then we'll kind of end off with some tools and resources. So actually, before we even say any of this, I want to say that cultivated meat is the most talked about within the EA community. And I, I think that there's actually a lot of tractability and neglectedness in the plant-based and fermentation spaces as well. I think it's interesting to think about why EA is kind of most excited about cellular agriculture. And I am going to focus on that today, but I want everyone to keep in mind the fact that like plant-based and fermentation also have a ways to go and have a tremendous opportunity to disrupt industrial animal agriculture, and we should not discount them as a community. And yeah, so we can talk about that more in the Q&A section if you guys want, but let's dive into the first core research area for cultivated meat. So the first thing that anybody needs to do research and make research advancements in this space is cell lines. You need the cells themselves. And yet there's a really massive cell line shortage. So some of the most important recent research advancements in cell line development, many of which were funded by GFI's own research grant program, is around cell banking. Can we essentially create a biobank of all of the agriculturally relevant species or all of the meat relevant species, and make these repositories open access so that anybody who wants to do research in this space won't have trouble sourcing cell lines. And one of the reasons these cell lines are so difficult to source is because we have lots of human stem cell lines and rat stem cell lines, but we haven't really used tissue engineering for the food space yet. And that's why this massive white space exists. And these are some of the things that we're working on right now to fill that white space. We also started a partnership with Carafast to create an open access cell line repository so that various researchers, whether working on cultivated meat right now or not, can contribute to this biobank and make sure those cell lines are available for other researchers. So once you have the cell lines, I'm gonna throughout also lightly cover basic science around alternative proteins. You also need to kind of feed these cell lines with nutrients, that's cell culture media. And you need to make sure those cells have something to grow around, that's the scaffolding. And then this entire process is housed in a bioprocess. So that's context I probably should have offered a couple minutes ago, but now focusing on cell lines, there are, in addition to banking cell lines, in addition to coming up with new methods for generating animal cell lines, there's also this question of, which starting cell line types should we be isolating and banking in the first place? Right? We need to actually decide which starting cell type across the source materials that include embryonic stem cells, induced pluripotent stem cells, mesenchymal stem cells, adult muscle stem cells. There's really no consensus right now in the industry around which of these starting source materials are best suited for cultivated meat companies. And there's really not even a consensus around what the advantages and disadvantages are of each. So potentially a white space that a student researcher might be able to help us bridge a gap on is doing an analysis of the various cell types and understanding what their advantages and disadvantages are, making that information public so that companies can be able to make these decisions and research labs can make these decisions with less duplicative effort. So that's one example, white space. I mentioned that once you have the cells, you have to feed those cells something. And that something is cell culture media, which is comprised of sugar, salts, amino acids, and growth factors, which are kind of signaling molecules that tell your cells how to behave. Do I want the cell to expand really rapidly and proliferate, basically double, or do I want the cell to now differentiate into its end cell type, a muscle cell or a connective tissue or fat? Cell culture media, and this was surfaced during our brainstorm earlier, is remarkably expensive. And that's because most of the cell culture media is pharmaceutical grade. And what we really need is cheap, animal-free, so serum-free cell culture media that is affordable and kind of consistent across batches. And there have been a number of recent advancements made in this technology area that I also want to surface here. So we definitely see recombinant growth factors being grown. In general, cell culture media, the entire field is moving toward using serum-free media. By the time cultivated meat is scaled, there's no company out there, to my knowledge, that's intending to use um, fetal bovine serum. So maybe this helps alleviate some of the concerns that we surfaced earlier around the ethics of cultured meat. The low cost cell culture media is one really critical opportunity where we've seen some advancements, but further advancements are needed. These are some of the active projects that we're funding around using novel techniques for optimizing media and reducing the cost of cell culture media. The Burridge Lab at Northwestern University recently were able to reduce the cost of cell culture media by 97%, even at the bench scale, to around $10 a liter by producing stable growth factor variants in-house. And there are additional interventions we can take in order to reduce the cost of cell culture media even further, down to maybe like $1 per liter. If you guys are interested in further reading, you can check out our analysis of culture medium costs and production volumes by Dr. Liz Specht. That's available on our website, and I can give the link afterward as well. But some example white space in cell culture media that we definitely want to be able to tackle. One example is the knowing that cell culture media presently accounts for 60 to 90% of the cost of cultivated meat and will likely continue to be the most expensive part of the cultivated meat production process. Can we recycle it? Can we build our bioprocess in such a way that we're utilizing our media as effectively as possible? And in the cell proliferation phase, making sure that, yeah, we're using media throughout, cleaning it, and then bringing it back into the system. And then a second one is like, can we make cell culture media for cultivated meat that is food grade rather than pharmaceutical grade, utilizing affordable raw materials or even agricultural side streams in order to source those amino acids and small peptides to form the basis of our cell culture media? That's also kind of a gaping white space opportunity for you all to take on. And one note about this is that there is an existing research project at NTU University in Singapore. We recently had a scientific seminar about this that's about utilizing spent grain to produce viable cell culture media for fermentation. So this has been demonstrated, and they are also exploring how we can create cell culture media for cultivated meat. Through that same process of utilizing waste streams from the existing agricultural industry. So once you have these cells and you fed these cells something and they're growing, you also need to make sure that there's a 3D structure for them to grow around. Namely that the structure is either edible or biodegradable and very porous so that the media can flow through and reach those cells. I think that the cells need to be, is it 10 microns away from a nutrient source in order to grow? That's like hair lengths away or hair's width away from a nutrient source in order to grow effectively. And so that's why it's so important to have these scaffolds built in such a way that almost mirrors our vasculature, right? Our blood vessels that are constantly transporting oxygen and nutrients and amino acids to the cells so that they can thrive and we don't experience cell death. So some recent advancements in this scaffolding, this technology that will allow us to create more sophisticated whole muscle cuts of meat, um, are first and foremost utilizing plant-derived materials. So can we use textured soy protein scaffolds, like Dr. Tem Ben Ayer, who works at GFI in Israel as my colleague? Can we utilize plant-based materials in order to create that kind of scaffolding? Tom used textured soy protein, which is like amazingly cheap, and widely available, and was able to successfully grow bovine skeletal muscle tissue on this material, because it's already kind of porous and structured in the way that we would want. And that formed the basis for the technology that kicked off Aleph Farms, to be able to grow these kinds of cultivated steaks. So utilizing plant-derived materials. Some other examples of this, at Glenn Gaudet's lab at Worcester Polytechnic Institute, they're experimenting with decellularizing plant materials like spinach as pictured in order to kind of seed cells onto this extracellular matrix, which is already constructed in such a way to facilitate nutrient transfer. So this is another example. A company called Ecovative is using mycelium, which is like mushroom, the roots of mushrooms, kind of very delicate fibers, in order to create scaffolds as well, for cultivated meat. And this is an example of their cultivated bacon. And then Marie Gibbons at Harvard Medical School also experimented with seeding turkey cells onto jackfruit. And this is kind of what you see pictured here, on a microscopic level. Some other research advancements in scaffolding include, some of these projects were also funding at the Technion, and at UCLA, regarding basically like creating maybe tunable scaffolds where you can control really carefully the mechanical properties of these scaffolds and customize them as needed, and create the right kind of marbling so you can utilize the scaffold itself to create the right layers of fat and muscle tissue and create the desirable, muscle fat marbling that we see in many steaks. She's using hydrogel scaffolds. And then there's also this exploration of like 3D printing, for cultivated meat design. Can we 3D print that scaffold, that then cells are seeded onto, in order to make this process more efficient? There's also been some experimentation around like electrospinning. So using sugars essentially to spin scaffolds. This was done at the bench scale utilizing Legos. It's one of my favorite examples of just kind of playful experimentation being able to yield potentially useful insights. And so I think the realm, this terrain of what kinds of materials we should use for our scaffolds, and what properties, how can we ensure optimal nutrient flow, remains like a massive white space. And then there's this potential example white space of developing computational models of perfusion flow. So for those of you that might have a computational sciences background, this is a white space for you to think about paired with someone with a biology background and a tissue engineering background. But essentially, what we see a great need for are models that describe the fluid flow through the scaffolds, and to help us better understand shear forces and mass transfer rather. Shear forces being those like mechanical stresses essentially on the cell because of the friction between cells as fluids pass around them that will oftentimes alter cell behavior and cell adhesion. So better understanding how this fluid flow will impact cell dynamics at scale, will do a lot in the way of informing considerations for what materials we should use and what geometries those materials should exist in, what form they should take, the dimensions of these scaffolds, the different kinds of methods we should use to fabricate those scaffolds, the entire bioprocess it will have very important implications for. And also, it goes back to the composition and the viscosity of cell culture medium. So these computational modelings, understanding how cells behave at scale and how fluid really flows through these scaffolds will kind of be critical for understanding the overarching cultivated meat production process. And so all of this, what I talked about, all of the entire process is housed in a bioprocess, an industrial scale bioprocess. And we've seen some recent advancements in bioreactor design and real-time online monitoring. So we've been funding some projects that utilize kind of custom bioreactors, trying to build, we have bioreactors from the fermentation industry, from the biopharmaceuticals industry, but they've not been optimized for cultivated meat production. So that's why it's so important for us to actually build bioreactors that are tailored to cultivated meat. And if we are able to kind of have these sensors for real-time monitoring that are kind of embedded in line, and people can see the researchers can easily track how cells are proliferating and differentiating, it can be easier to basically guide the cells, the entire batch of cells at industrial scale, and guiding them to differentiate into the desired cell types at the right time. But of course, like the other core research areas, research advancements or an example of a white space opportunity that still remains really underexplored is maybe this designing open access blueprints for facilities. So there's a lot of redundant effort and missed opportunity that we're experiencing in the cultivated meat space because we haven't yet gone to take the step of identifying what facility improvements can be made across the entire industry. So is there an opportunity here for some of you maybe, potentially with a chemical engineering background, bioengineering background, electrical engineering background, an industrial manufacturing and design background, to put your minds together and build a set of open access blueprints that could provide a head start on facility design, which would really allow equipment manufacturers and engineering companies to become more familiar with the industry needs, even though ultimately these facilities will need to be customized likely to each company. There's this opportunity right now way before any of these companies are producing at industrial scale to start getting adjacent industries thinking about what facilities like this will look like so they can start to meet industry needs. So a few call to actions. There are so many more white space ideas that exist than I could cover in this talk. We've really barely touched the surface. A really key call to action for any of you who are interested in continuing to understand research advancement in this space and who want to explore additional white space opportunities, you should join our scientific seminar series at gfi.org/community. In the last 6 months, these were some of the topics that we covered, and you can also watch past seminars. And up next, we have just next week, in 3 days on Wednesday, we'll be joined by Dr. Julian McClements, who will dive into the science behind making much better plant-based milks and closing the sensory and functional gap. And then the following month, we will be talking about developing a scalable bioprocess for cultivated meat production with GFI grantee, Dr. Petra Henga. I also want to draw everyone's attention to gfi.org/alternative protein solutions. because this is a resource that we've only beta launched so far. Don't disseminate it too widely, but it basically gives you a solution database of all of the white space ideas. We're releasing them in batches, but additional white space ideas that we have generated along with a panel of 150 industry experts that we think are kind of crucially needed to fulfill in this space. And those are research ideas, there's company ideas, those are ecosystem level interventions, and each of them come paired with these opportunity briefs or concept notes so that you can read more in depth. And there will be an engagement forum on the site as well, so that if you do want to work on any of these ideas, let us know. We can help you find the right collaborators and make sure that you have the resources you need to mobilize these white space opportunities. So that's all that I wanted to cover today. Make sure you visit gfi.org. Make sure you email me at amyh@gfi.org if you have specific questions that potentially I can help answer around some of these white space opportunities and around what you as students can do to close some of these white spaces. But I will stop sharing my screen at this point and open it up for general questions. Thanks everyone for listening.